We all start as a single cell, and then 2, and then 4, and then 8, and then so on until you're a whole person. But those early cells have to do more than just multiply. Eventually, they also have to differentiate into different kinds of cells. So how do scientists know which of those beginning cells turn into the different parts of you and when those critical branch points happen? Let's talk about it.
Okay, so imagine that you are an ancient monarch and you need to get a message out as quickly as possible to every single member of your kingdom, but without speaking a single word out loud. So you write out 2 copies of the message, you hand them each to one of your friends, you tell them each to write down 2 more copies and hand them to each of their friends, etc. The message will spread exponentially as people copy it out by hand and pass it on. But inevitably there will be some mistakes. By the time everybody gets a copy of the message, there may be thousands of copies floating around that have accumulated dozens of mistakes. And by examining the trail of mistakes, you can trace back to the point at which each of them happened. This also happens in your DNA. Every time your cells replicate, your DNA has to be duplicated so that each resulting cell has a full copy. And whenever you're making copies, mistakes can happen. The good news is most of the time these mistakes turn out to be harmless, and scientists can trace these subtle genetic changes back to figure out which cells originated from which. It's called quantitative fate mapping via retrospective lineage barcoding, which is just a fancy way of saying the following. Gather up all the copies of the message, or DNA, from all the cells of the body, find the differences between them, and arrange them in a tree structure according to those differences until you get back to the original. That way you can easily trace out all of the branching paths.
Okay, to be clear, we haven't actually gone like all the way back to egg cell with this yet, but we have shown that this is a very effective method to go from progenitor to terminal cell for a lot of different kinds of cell types.
This gives us really important information about how people get made. We all start as one cell, and most of us make it to 30 trillion, and all of those 30 trillion cells have a job to do. Except for your appendix cells. As far as we know, they're just in there, like, not paying rent, getting zooted, watching the tuning exams arc for the millionth time. But the cells in your eyes and your brain and your intestines and your lungs and your kidneys and your skin, they all have a job to do. They're working so hard, and they all came from one single fertilized egg. And fate mapping is an incredibly powerful tool that helps us figure out the incomprehensibly complicated and quite underratedly beautiful journey of how we get from here to here.